Module 2, Plant Physiology, Bryophyta to Gymnospermy, Protozoology, Arthropoda, and Mollusca. Lecture 1, Plant Physiology I. Plant physiology explores the functioning of plants, focusing on growth, development, and metabolism. 1.1 Seed morphology and germination. A seed consists of embryo, the miniature plant; endosperm or cotyledon, nutrient reserve; seed coat, protective layer. Germination begins when a viable seed absorbs water, imbibition, and resumes metabolic activity. The process includes activation of enzymes, cell elongation and division, emergence of the radicle, root, and plumule, shoot. Factors influencing germination: water, oxygen, temperature, and sometimes light. Herb 1.2 Growth patterns, from seed to senescence. Plant growth is indeterminate in many organs, e.g., roots, shoots. Determinate in organs like leaves and flowers. Key stages: 1. Germination. 2. Vegetative growth, root, stem, leaf development. 3. Reproductive growth or flowering, fruiting. 4. Senescence, aging and death. Growth involves cell division or mitosis at apical meristems. Cell elongation. Differentiation into specialized tissues. Blossom 1.3 Flowering and fruit formation. Flowering is triggered by photoperiod, length of day/night. Hormones, e.g., gibberellins. Fruits result from fertilization of ovules, which develop into seeds, while the ovary becomes the fruit. Seed and fruit dispersal mechanisms: wind, water, animals, mechanical ejection. Deciduous tree 1.4 Growth factors and analysis. Internal factors: hormones, auxins, gibberellins, cytokinins, abscisic acid. External factors: light, water, temperature, gravity. Growth analysis methods: measuring biomass, calculating leaf area index, LAI, assessing relative growth rate, AGR. Studio Microphone Lecture 2, Bryophyta, Pteridophyta, and Gymnospermy. Herb 2.1 Bryophyta, mosses and liverworts. Non-vascular plants. Found in moist, shaded areas. Dominant gametophyte generation. Require water for fertilization. Bryophytes include mosses, e.g., Phaneria. Liverworts, e.g., Riccia. Hornworts, e.g., Anthoceros. Ecological roles. Pioneer species. Prevent erosion. Bioindicators of pollution. Seedling 2.2 Pteridophyta, ferns and allies. First vascular plants, xylem and phloem. Dominant sporophyte generation. Reproduce via spores, not seeds. Examples: ferns, e.g., Nephrolepis. Club mosses, e.g., Lycopodium. Significance: some are ornamental. Indicators of forest health. Source of bioactive compounds. Evergreen tree 2.3 Gymnospermy, conifers. Seed-bearing plants without fruits. Vascular and woody. Dominant in temperate and boreal regions. Examples: pine, pinus. Cycads. Ginkgo. Features: naked seeds. Pollination by wind. Secondary growth, wood and bark. Gymnosperms provide timber, paper pulp, resins, and medicinal compounds. Microscope 2.4 Origin of vascular elements and seed habit. Vascular tissues, xylem and phloem, evolved for efficient transport. Seeds offered protection and survival advantage over spores. Seed habit evolution. Gametophyte reduction. Development of ovule. Pollination mechanisms. These features allowed plants to colonize drier habitats and become dominant on land. Studio Microphone Lecture 3, protozoology. Protozoology is the study of protozoa, single-celled eukaryotic organisms found in water, soil, and as parasites. Microscope 3.1 General classification on features. Protozoa are classified into Sarcodina, e.g., amoeba, moved by Pseudopodia. Mastegophora, e.g., Trypanosoma, moved by flagella. Coleophora, e.g., Paramecium, moved by cilia. Sporozoa, e.g., Plasmodium, non-motile, parasitic. They have contractile vacuoles, complex feeding mechanisms, diverse reproductive modes, asexual and sexual. Microbe 3.2 Parasitic protozoa of medical importance. Examples: Plasmodium spp. Cause malaria. Trypanosoma spp. Cause sleeping sickness. Entamoeba histolytica causes amoebiasis. Giardia lamblia causes giardiasis. DNA 3.3 Modes of infection, life cycles, and control. Infection by ingestion, vectors, mosquitoes, tsetse flies, or contaminated water. Complex life cycles involving multiple hosts, e.g., Plasmodium alternates between humans and mosquitoes. Control measures. Improved sanitation. Vector control. Antiprotozoa drugs. Public health education. Studio Microphone Lecture 4, Arthropoda and Mollusca. Cricket 4.1 Arthropods. The phylum Arthropoda includes insects, crustaceans, arachnids, and myriapods. Key characteristics: segmented bodies, exoskeleton made of chitin, jointed appendages, open circulatory system. Classes include Insecta, beetles, butterflies, mosquitoes. Arachnida, spiders, ticks. Crustacea, crabs, shrimps. Ant 4.2 Internal and external structures. Systems: digestive, complete, with specialized organs. Excretory, Malpighian tubules, insects. Respiratory, tracheae, insects, gills, crustaceans. Circulatory, open system. Nervous, ventral nerve cord and ganglia. Reproductive, sexual, with internal or external fertilization. Adaptations include camouflage, flight in insects, complex social structures, e.g., ants, bees. Spiral shell 4.3 Mollusca. Phylum Mollusca includes soft-bodied animals, most with calcareous shells. Classes include Gastropoda, snails, slugs. Bivalvia, clams, oysters. Cephalopoda, octopuses, squids. Features: muscular foot. Mantle that secretes the shell. Radula, except bivalves. Closed, cephalopods, or open, others, circulatory systems. Gear 4.4 Comparative morphology, development, and ecology. Mollusks show bilateral symmetry. Development includes larval stages, e.g., trochophore. Adaptations: shells, ink, cephalopods, jet propulsion. Roles in ecosystems: filter feeders, decomposers, prey and predators. Economic importance: food, shellfish. Pearl industry. Environmental indicators. White heavy checkmark Module 2 Summary. In this final module, we have studied 1. Plant physiology from seed germination to flowering and fruiting. 2. Morphology and reproduction of Bryophyta, Pteridophyta, and Gymnospermy. 3. Life cycles and classification of protozoa and diseases they cause. 4. Anatomy, physiology, and ecological roles of arthropods and mollusks.